Everybody, my name is Vijay Tiwari. Today I'm going to tell you about how single-celled RNA sequencing methods have begun to impact discovery as well as health sciences. Single-cell technology was named Breakthrough Technology of the Year by Science magazine in 2018. Since then, it has opened another door to life science research, and scientists around the world have entered the fast track to uncover the mysteries of life. In 2019, single-cell multiomics was named Technology of the Year by Nature Methods. In 2020, the Technology of the Year Nature Methods was spatial transcription technology that complements single-cell transcriptome sequencing in a way that it looks at the gene expression in a 3D context. Single-cell technology is working wonders and witnessing history time and time again, and it has started to influence clinical decision making in many different ways. If I have to give you an example, you could imagine a complex fruit salad. If you will grind all this together as a juice, in the end, if the juice tastes bad, you would not know which of the components was bad. And that's the beauty of single-cell analysis methods. Here, we look at one fruit at a time. So in the end, you would get to know what was the problem if you would have mixed them together. So this is a good example because every tissue is complex. It consists of many different cell types. So in the end, if you have to identify different types of cell populations, that's the way to go. And that's further exemplified here. For example, here a single patient presents with cancer at multiple sites. If you zoom into one of these sites, we notice both neoplastic and non-neoplastic cells. Within the neoplastic cells, we see two different distinct subpopulations marked by different somatic mutations, for example, indicated with a star here, for example. Likewise, within non-neoplastic cells, there are different types of T cells and B cell populations, as well as other cell types. Bulk measurements provide the average quantification of gene expression and likely average all of these profiles, which potentially obscures proportion and subpopulational stage-specific differences. Hence, these consequences are visible when you plot the bulk data versus single cell data. So in this case, if you have a complex heterogeneous tissue, you will analyze different cell types one cell at a time. In the end, you would be able to create gene expression profile for each single cell, and you could cluster them based on similarities in their expression profile. And typically, the cells that cluster together, they create a subpopulation, often what we call cell type. If you would have mixed them together, you would create an average gene expression profile that does not really reflect or is able to show problems occurring at the single cell level at many different cell types. The workflow for single cell analysis is very simple. You take an intact tissue, you dissociate, create single cell suspension, and depending on what kind of single cell analysis you want to do, either DNA analysis, RNA analysis, or DNA methylation, you would create different types of libraries. In the end, the sequencing libraries are created, these are sequenced, and then following data analysis, one is able to create this subpopulation of cells based on single cell DNA, RNA profiles, and so on and so forth. The most popular method of single cell analysis at the moment is 10x genomics. In this case, we isolate cells and we could ship the cells anywhere in the world. The first step before beginning 10x is removing the dead cells. We need highly viable cells in this protocol. So the cells are then processed by what is called 10x genomics chromium to create single cell libraries. The libraries are then pooled on the Illumina platform. Within this chromium controller, the cells are loaded onto a microfluidic chip and encapsulated within droplets containing barcoded gel beads and reagents for reverse transcription. Following cell lysis, the beads capture the poly A tails of the mRNA molecule. Reverse transcription generates cDNA tagged with a 10x barcode to identify the cell and a UMI or unique molecular identifier to label the mRNA transcript. The pooled cDNA is amplified in bulk, and Illumina adapters including a sample index are added to the fragments to generate sequencing libraries. There are a number of applications. For example, As shown earlier, you could take a heterogeneous tissue. In this case, it was a primary visual cortex. This visual cortex was dissociated, and approximately 1,600 cells from this visual cortex was subjected to single-cell RNA sequencing. An unbiased analysis first reveals the presence of neuronal versus non-neuronal cells, again based on the presence of certain markers. The non-neuronal cells further segregate into different glial types as shown here. And that in that case, those glial cell types would be able to give you different profiles, and same was done for the neuronal cells. In this example too, we are looking at a heterogeneous pancreatic ductal adenocarcinoma cells. As you can see, the cells are flooded with different types of tumor cell types. Here we have fibroblasts, we have different types of tumor cells, we have immune cells, we have circulating tumor cells. So in order to understand which of these are contributing to the aggressiveness of the tumor, how we can use this for therapy, one is able to isolate all of these cell types and then analyze them. This is a third example. In this case, one could take tumors from patients and analyze for their epigenome profile or transcriptome profile or proteomics. And based on the information we would collect for each of the cell types within a tumor, one is able to create personalized therapy or create biomarkers for disease progression and therapy resistance. In this case, one is able to see 4C clinical trials guided by single-cell RNA sequencing, either by taking a tumor biopsy or peripheral blood cells. One is able to perform single-cell RNA sequencing, TCR sequencing, and so on and so forth, or guide immune therapy, for example, linking single-cell RNA-seq to bulk whole exome and by bulk RNA sequencing, and eventually performing high-dimension analysis to guide immune resistance. With the examples, I hope I have convinced you the tremendous power of single-cell RNA sequencing technology and the potential it holds to make breakthroughs in the area of health data science. Thank you very much.